harvesting complexes and model systems. So Jessica, the screen is yours.
Let me see. Let me share my screen. Okay, and let me move this here. Okay. All right, the pointer. Here we go. Okay. Everything's good. All right, well, thank you very much. I wanted to first thanks to Thomas and Maxime for really organizing these conferences and for giving me the opportunity to tell you about some of the work that we're doing in my group. So we have a few different projects that we're working on, but these projects have a common theme. Oh, hold on. There we go. Okay, so these projects have a common theme, and that's light harvesting. So here's a schematic for photosynthetic light harvesting, where you'd have different chromophores, they absorb photons, transfer this energy to a reaction center, and then you have charge separation taking place. And this can happen with a really high quantum efficiency. And this is common for all these different photosynthetic organisms. But what's really interesting is when you start looking at the main light harvesting complexes, you can see they're very structurally different. So here's light harvesting complex 2 from plants, and here's the main light harvesting complex from purple bacteria. Right, so they have different structures, but they're performing the same function. And so researchers in this area are really trying to understand what are the parameters that lead to this robust and efficient light harvesting. So what's important about the geometries of the relative chromophores? What about the role of vibrational motion and energy transfer and charge separation? And what about interactions with the local environment? And we heard some really nice talks previously at these meetings talking about different light harvesting complexes and looking at these different aspects. And so my group also focuses on this, is motivated by these problems. And the natural light harvesting complex we look at is shown here. So this is photosystem 1. We're trying to understand how energy flows among these different chlorophylls, how it reaches the reaction center, which is here, and ultimately how charge separation takes place. And we're making progress here, but photosystem 1 is a complicated system. So along with looking at photosystem 1, we're also looking at artificial light harvesting chromophores and isolated cofactors. So here you can make structural changes to the molecule, and you can make systematic changes to the bath, right? You can change the solvent. And we can see how those changes affect the properties of the molecules, but we can also see how that information presents in the 2D spectra, and that helps us with interpretation. Right, and the third area of research that we're working in, I call it bio-inspired systems and photocatalysts. These could also be considered model systems, but I classify them differently because they can do chemistry, right? So we have a nice collaboration with Eric Shelter's group looking at these cerium-based photocatalysts. We have a collaboration with Neil Thompson looking at these copper-based systems, which are copper enzyme mimics. And here we're really trying to understand how altering the properties of these ligands here can influence the ultrafast dynamics at the metal centers, and ultimately, can we link this to reactivity? The other area that we're working on are these metal carbonyl complexes. So these systems are well loved by this community, right? And so we're also studying these systems, but we're looking at them in self-assembled cages. So we're trying to really understand how non-covalent interactions and spatial confinement can alter the reactivities of these carbonyl complexes in these cages. Okay, so to study these systems, we use ultrafast spectroscopy, we use pump probe spectroscopy, and also two-dimensional spectroscopy. And because this is CMDS, I'm going to talk about our work using 2D spectroscopy. And to obtain the 2D spectra, here's a picture of our laser lab. So on this side of our laser table, we built a 2D electronic spectrometer, so we can really look at energy flowing among different electronic states. On this side of our laser table, we built a 2D IR spectrometer, so we can really look at dynamics on a ground electronic state. And we built these spectrometers on the same laser table so we can start mixing the pulses. So we're now obtaining 2D electronic vibrational spectroscopy. Today, I'm going to focus on our work with 2D electronic spectroscopy. Okay, so you're probably wondering why I gave you sort of an overview. I'm this an assistant professor at Penn, so I thought it'd be good to let you know what we've been up to these past few years. And now I want to tell you 2 stories. Right, so the first is going to look at photosystem 1. Right, so here's the photosystem 1 drives oxygenic photosynthesis. It's found in the photosynthetic membrane, but we're going to look at isolated PS1 complexes. And here's the structure. So it looks like a clover, all right? And each leaf of this clover is a monomer. And at the center of each monomer is a reaction center. And so here's another view of the reaction center. So these iron sulfur clusters are these yellow balls here. Okay. So most of these chlorophylls, the chlorophylls are in green, are act as antenna. And most of the antenna chlorophylls are bulk chlorophylls. And I cut the tails off the chlorophylls so it doesn't get too messy. So they just look like porphyrins. But, okay. So most of these chlorophylls are bulk chlorophyll. They transfer energy downhill to the reaction center. But what's interesting about photosystem 1 is that it also has what's called red chlorophylls. And these are all chlorophyll A molecules, but they lie in they have such a geometry that their lower lying excitonic state lies to the red of the reaction center. Right? And so in order for these red chlorophylls to contribute to charge separation, they have to undergo an uphill energy transfer event. And there's, right, so this is the general schematic for light harvesting, right? We know a lot about this, but there's still unanswered questions regarding sort of energy transfer and charge separation in this system, and specifically focusing on these red chlorophylls. So how are the red chlorophylls connected to the other antenna chlorophyll or the reaction center? What's the identity of the red chlorophylls? So there's still sort of questions to which chlorophylls are actually the red chlorophylls. And there's also questions regarding the initial charge separation event. And so there has been a lot of studies here. I'm just highlighting some reviews. And so I guess one question is why are there still unanswered questions? And that's really for two reasons. First is there's a lot of spectral congestion here. So here's the linear spectra of photosystem 1 from 3 different cyanobacteria. And all of the chlorophylls contribute to this QY band here. And even the charged species like the anion and cationic form have absorptions here. So we see a little bit of structure associated with the red chlorophylls, and these are species dependent, but there's still a lot of spectral congestion. There's also overlap of temporal processes. So in photosystem 1, you can't actually decouple the reaction center and the light harvesting antenna. So you have all of these chlorophylls sort of stuck together. And so you have energy transfer occurring among the antenna chlorophyll and the reaction center chlorophyll, and also the primary photochemistry occurring on the same time scale. So what we're doing in my group is applying 2D electronic spectroscopy to photosystem 1 to try to alleviate some of the spectral congestion, to see if we can get some more insight into energy transfer in the system, specifically focusing on these red chlorophylls here. Okay, so we have today I'm going to talk about our work on 2 different PS1 complexes. So, and they were chosen because they have different red chlorophyll content. So PS1 from 7002 has 1 red chlorophyll. All right, so we're going to call this one the one with less red chlorophyll. And PS1 from 6803 has 2 pools of red chlorophyll. And we're going to call this one the one with more red chlorophyll. Okay, so you can see more of an absorption. This is a collaboration with John Goldbeck's group at Penn State University, where they're sending us these PS1 complexes. And this was work done by former graduate student Yumin Lee. She's now a postdoc in the Tokmakov group. Okay, so before showing the 2D spectra, I just wanted to sort of look at these schematics to help us interpret the spectra. So let's pretend that we could actually resolve a peak for the bulk chlorophyll, resolve a peak for the reaction center, and resolve a peak for the red chlorophyll. Then at early times, we would expect to see transitions on the diagonal associated with these peaks, right? As the waiting time increases and energy starts to flow among these different states, we could see that as an appearance and growth of cross peaks. So for example, this peak here would indicate excitation of the bulk, but detection at the red. So it would indicate downhill energy transfer to the red chlorophyll. Okay, so let's look at a more realistic schematic spectra. We can't actually resolve these different transitions, right? And so our early time 2D spectra will look it'll have a lot of static heterogeneity, right? So we'll have this sort of elongated peak. And as the waiting time increases and energy starts to flow among these different states, we won't see clear cross peaks, but we can see amplitude growths in these cross peak regions. So we can still characterize the energy transfer. And this is more realistic or more representative of what the 2D spectra look like. Okay, so here are the 2D spectra. So we have photosystem 1 with more red chlorophyll on the top, photosystem 1 with less red chlorophyll on the bottom. And I'm plotting excitation axis here and detection axis here. And in the corners, I denote the waiting time. All right, so to interpret the spectra, I'm going to draw lines. All right, so here's excitation of the bulk antenna. Here's detection at P700, and this would be detection at the red chlorophylls. So this is our cross peak region here. And we see at early times, there's little amplitude in this cross peak region, right? As the waiting time starts to increase, we see more amplitude growth in the cross peak region. And it's more clear if we look at the traces. So for example, I'm plotting the trace in green here is on the diagonal, we see this decay. and the traces taken at these coordinates here in the cross peak region, we see a growth followed by a decay. And this peaks at about 9 picoseconds or so. Okay, so we can do the same thing with PS1 with less red chlorophyll, right? We can draw the lines. And we see the same general trend, right? Little amplitude here at early times, and then at later times, we see a growth in this region. And so if we look at the traces, right, we see an amplitude growth for the red chlorophyll, followed by a decay. And this peaks at maybe about 3 picoseconds. Okay, so this is from this spectra, right? We're getting some information that there's downhill energy transfer to the red chlorophylls. We can see this as sort of a growth. But what we'd really like to do is try to extract some more information from the 2D spectra, right? So we have this sort of line shape change. Can we go back to sort of this picture about of the connectivity? All right, and so to do this, what we're going to do is use a compartmental model based off of previous pump probe spectroscopy on photosystem 1, where we have the bulk antenna, we can have red chlorophylls, we can have a reaction center and the radical pair. And then we're going to apply a global analysis to the 2D spectra. And there's been a lot of previous studies sort of establishing how to do this and the power of global analysis for interpreting the 2D spectra. Okay. So I'm not going to go over this slide that much, but I just want to say that when we think about the global analysis, we can think about the 2D spectra as a sum of n components, where each nth component has an amplitude, and the amplitude evolves with the function of, or evolves with this time scale Tn. And so this amplitude here, these are going to be the 2D decay associated spectra, and I usually end up calling these 2D DAS. Okay. So before looking at the 2D DAS of photosystem 1, I just want to look at this model system here because we're going to use the spectral patterns to help interpret the spectra of photosystem 1. All right, so this is a simple model system. We have two states that can undergo equilibration and then decay to the ground state. And what we did is we just wrote a series of rate laws, and we can solve these rate laws to, or the coupled differential equations to predict how the amplitudes of these peaks will evolve as a function of time, right? So we see that the diagonal peaks are going to decay, the cross peaks are going to grow in and then decay. And then this is a really simple model because then what we do is we take 2D Gaussians and we just weight them by these amplitudes. So we can predict sort of how the population changes to the 2D spectra. So there's no line shape information here. Okay, so then what we do is apply a global analysis to the spectra here, and we can extract two components. And here are the two components. And to interpret these, the negative peaks mean decay and the positive peaks mean a growth. And so for our model system, right, we see that on the 20 picosecond time scale, all of the peaks in the 2D spectra decay. And that makes sense, right? That's our lifetime, that's what we put in, so we should get that out. Right, now if we look at this faster component, we see that the peaks, this component's evolving on the 2 picosecond time scale. And if we look at the pattern here, we have the diagonal peaks are decaying and the cross peaks are growing in. And so this is the time scale for equilibration, right? Given these uphill and downhill time constants or rate constants. So we have a equilibration, and we have this clear pattern for equilibration where we have these 4 peaks. So we have this box pattern. And from the position of the peaks in this box, we can extract sort of the transitions of the states involved in the equilibration process. Okay, so now we'll come back to this in looking at PS1 data. All right, so we applied the global analysis to photosystem 1. We determined there's 5 components. This is consistent with previous work. And here are the 2D decay associated spectra. So I'm going to walk through these, but here's more red chlorophyll, less red chlorophyll. The spectral patterns are similar, so I'm just going to focus on one data set now. All right, and we want to assign the 2D DAS. So for the assignment, what we're going to do is look at the spectral patterns. We're going to look at the timescales. We're going to think about this in the context of previous studies. We're also going to compare this to our model spectra. And remember, the goal here is to try to think about the connectivity of these different compartments. Okay, I want to tell another story today, so I'm going to just focus on the results for the red chlorophylls. All right, so here on the 4 picosecond time scale, okay, we see this is the 2D DAS for photosystem 1. So we have this box pattern here, right? And if you remember, it's the same box for equilibration. So this indicates there's equilibration occurring among these states. And from the position of the cross peaks, we have we know that this involves equilibration with the red chlorophylls and lower lying antenna states or relaxed antenna states, right? Because this is shifted from the diagonal. Okay, so we can sort of draw these arrows. And this is consistent with previous studies. Okay, now we can look at the 200 femtosecond component. So now we have two boxes here, right? And so we have two equilibration processes. And just to check that, we made a model system to see if that checks out. And from the position of these two boxes, we find that there's equilibration among the higher lying antenna states and lower lying antenna states. In addition, we see this equilibration involving the red chlorophylls, but now it's sort of on the diagonal. So it's either involving other antenna states that are not relaxed or possibly reaction center states. Right, and this is also consistent with previous pump probes studies. But now we can gain a little bit more information, right, from the 2D DAS, because now we have this sort of connectivity map. So for example, we know that this antenna state here is not undergoing equilibration with the red chlorophylls. Okay. And then we can also resolve a very fast component that evolves on the 50 femtosecond timescale. And here, what we see is it just looks like a growth. So we have a growth that's sort of slightly shifted below the diagonal. And there's been previous studies on other light harvesting complexes that have fewer chlorophyll molecules. So with those studies, cross peaks can actually be resolved. Right, so you can see cross peaks with excitonic states, and you can see a growth that's on the 50 femtosecond timescale, and that's been attributed to exciton relaxation or relaxation within an excitonic manifold. Right, and so that could be giving rise to this peak here in photosystem 1, but there's also some possibilities, other possibilities too, right? Because we can't resolve, there's lots of different chlorophylls contributing to this peak here, so we don't resolve individual cross peaks. And some of the other possibilities is there could be a dynamic Stokes shift contribution here, but there also could be vibrational relaxation. And I'm going to come back to the dynamic Stokes shift in the second part. Okay, so to summarize, we were able to go from this picture of the 2D electronic spectra of photosystem 1, looking at this sort of cross peak region here, the amplitude growth, to apply a global analysis to extract these 2D DAS, and then use the information contained here with model systems to really think about the connectivity of these different states. Okay, and then we can compare the time scales that we extract for PS1 with more red chlorophyll to the one with less red chlorophyll. And so I'm denoting those with like these underscores here. So we see on the ultrafast time scale, the relaxations are similar. The femtosecond energy equilibration has a similar time scale. But once we get to this picosecond energy equilibration, that's where we see the differences. And the PS1 with less red chlorophyll has a faster equilibration time. And so to think about this a little bit more, we can think about this through Forster theory, where the time scale for equilibration is proportional to the coupling and the spectral overlap. So this is a cartoon schematic, right? We don't know the actual spectra here, but in thinking about the energetics of the two red chlorophyll pools in the photosystem 1 complexes, the 706 and 708 nanometer pools lie closer in energy, and so we'd expect them to have a similar spectral overlap, so a similar time scale for equilibration. But the 714 nanometer pool in the PS1 with more red chlorophyll could have less, it's separated more in energy, right? And so it could have less spectral overlap. And so that would be consistent with this observation that we're seeing with a faster time scale. However, this is only one part of this expression. There's also the coupling, and the coupling is how the geometry enters this equation. Right? And so if we look at the structure of photosystem 1, so here's the structure of photosystem 1 from Tianlin-Gaddis, where the possible red chlorophyll candidates are highlighted in red. And more recent work by the Maser group have suggested that these chlorophylls here highlighted with ovals are the red chlorophylls in 6803. And so our 2D spectra, from the 2D spectra, we really can't say which chlorophylls are the red chlorophylls, but now we have the 2D ES spectra, and we have these really clear 2D DAS maps that can really serve as constraints for refining structure-based models where different possible red chlorophyll candidates can be tested, and then the resulting spectra compared to experiment. Oops. Okay, so that's our published work on photosystem 1. And now I want to shift gears a little bit to talk about some of our studies looking at line shape changes in 2D electronic spectra. Okay, so we all know that 2D spectroscopy is really a powerful technique for investigating solvation dynamics, right? And that's because these 2D line shapes here can be related back to these frequency fluctuation correlation functions. And through this correlation function, we can get information on solvent interactions. And there's a lot of different analysis procedures to extract solvation dynamics from 2D spectra, right? And here I'm just listing them. And they look at different aspects of this line shape change, but they all have something in common, and that's their relationship to the correlation function. But what about 2D electronic spectra of systems with larger Stokes shifts? Okay, so here's, in this scenario, the Stokes shift will lead to an elongation along the detection axis, right? And so to sort of consider that a bit more, like why would see the shape, we can look at this cartoon here, where excitation would be, this would be like the absorption, your absorption maximum. And then once you excite your molecule, it's going to relax, your solvent's going to reorganize, and that's going to lead to a decrease in your energy gap, right? So this is your Stokes shift. And this leads to a red shift here along the detection axis. You can time resolve this, right? If we can look at how this frequency is changing as a function of time, you can time resolve the Stokes shift, and that's the dynamic Stokes shift. And there's been a lot of previous studies looking at the dynamic Stokes shift. So sorry, these citations should be over here, but I'm running out of room, so I'll just connect it with this pointer. Okay, so there's been a lot of previous studies looking at the dynamic Stokes shift, and that's because the Stokes shift function here, where the parameters are defined on this cartoon, can be related, the Stokes shift function can be related back to the frequency fluctuation correlation function, right? So we have another reporter for solvation dynamics. And so in this sense, the dynamic Stokes shift can be another technique that we can use to extract solvation dynamics from 2D electronic spectra. Right, and so today I want to talk about our two recent studies extracting the dynamic Stokes shift from 2D spectra. First, focusing on Bo-Dippy chromophores, where we sort of show a procedure to extract the dynamic Stokes shift, and then looking at crescent violet molecules, where we're expanding on this procedure to account for strong oscillatory components. Okay, so I'll focus on the Bo-Dippy work first, and this is work that again was done by Yumin Lee, and our collaborator is Dave Chenoweth, so he makes different types of Bo-Dippys for us. Okay, so here's the 2D spectra of Bo-Dippy at 3 different waiting times. Here's the absorption spectra, and I'm indicating the maximum in absorption here. And green is the emission, and here's the maximum in emission with this dashed line, right? And we tune our incoming pulse so that we can overlap with both the absorption and emission. I guess that's key for these experiments. Okay, and what we see is as the waiting time increases, right, the peak becomes more elongated along the omega-3 axis. And this is the dynamic Stokes shift. And what we want to do is extract the Stokes shift function, right? So we can, so to do this, we need to think of how we're going to extract how this frequency here is changing as a function of time. So we're going to use 2 different analysis procedures here. First, we can project the 2D spectra onto the omega-3 axis, right? And get back our pump probe spectra. So we can sort of analyze this in the pump probe spectra with the projection, or we can take slices at different excitation frequencies and look at the solvation dynamics from a single slice in the 2D spectra. All right, so this side, so how do we actually do this? So here in blue is a representative slice or projection, okay, from the 2D data for a given T2 time. And so what we do is we're really looking at this dynamic line width change. And so we're gonna, for every projection as a function of T2, we're gonna extract the frequency at the full width half max. And then that becomes one of the points in this plot here. And so here we can we're going to take this as our nu of T. All right, so we're taking this as sort of our frequency that corresponds to the energy gap change. And then we're going to plug this into the Stokes shift function. Right, and so here are the results for Bo-Dippy, the one that we're looking at here, it's shown here. This is for the projection, and this is for taking slices at different points in the 2D spectra. And we see a decay, right, on a fast decay and then a slower decay. And remember, this is like the correlation function, right? And so we can extract the time scales. And what we observe is there's a 50 femtosecond Gaussian component. So we fit this with a Gaussian. And there's a longer 1 to 2 picosecond component. So we don't fully characterize the longer component because for these experiments, we only scanned out to about 2 picoseconds. Okay, so we're going to focus on the 50 femtosecond component, the ultrafast component, and we assign this to the inertial response based off of previous studies. And I should say that the solvent here is methanol, right? So previous studies on methanol. And the way to think about this, right, is our BODIPI, we excite our BODIPI molecule, we have this change in charge distribution, and the methanol molecules that are close to the BODIPI are going to have this hindered rotational motion, right? So that's this inertial response. That's what we're sort of thinking of measuring here. And it's consistent with previous studies of other chromophores and methanol performed with different techniques. And so we take this as a confirmation of our analysis procedure. Okay, so from this study, we were able to extract the dynamic Stokes shift from the 2D electronic spectra. And we were really excited about this because now we can extract solvation dynamics for a single slice in the 2D spectra. And that could be really powerful for thinking about 2D electronic spectra of multi-component systems. Okay, so then what we did is we expanded on this work. So we wanted to say, well, what if our system gets a little bit more complicated and we have really prominent vibrational coherences? How does that affect the analysis procedure or the solvation dynamics? And so for these studies, we're looking at crystal violet. So there's been a lot of previous work done on crystal violet, and that's because it does have these really nice oscillatory components, right? So it's a really nice system to test and confirm that an experiment's working well. Okay, so this is work done by graduate student Xiaowei Liu. And so here are the 2D spectra of crystal violet now. And we see longer waiting times, right? I guess it's key that we tuned our incoming laser pulses. So we're probing both the ground state bleach and stimulated emission, right? So that's key. So at later waiting times, we see this elongation along the diagonal, and that's the dynamic Stokes shift. In addition, with crystal violet, we see these really strong oscillatory components. So here I'm plotting the amplitude at these 3 different coordinates here, and they're color-coded. And so we see these oscillations. We can Fourier transform these oscillations to extract the frequency of the oscillation. So again, here it's color-coded. We can perform this analysis for the entire 2D spectra so that we can extract all of the oscillatory components contributing to the spectra. And that's this average spectra here in blue. And then what we do is we can compare these transitions to those calculated from DFT and TD-DFT calculations. So the key here is that we have these oscillatory components, we assign them to vibrational coherences, and they're really well characterized. Okay. So now we can extract the Stokes shift function. So I'm going to present the projection first. So here's the Stokes shift function from the projection here. And we see there's ultrafast relaxation, but there's also these oscillatory components. And it's not surprising, right? Because we have the oscillations in the amplitude. But the key here is they're really well characterized. So we can incorporate them into the analysis procedure and still extract the solvation dynamics. So we can extract the inertial solvent response of about 50 femtoseconds. Okay, so then what we wanted to do was move on to the slices. And so we're going to extract the Stokes shift S of t from the slices here. And we're going to look at slices taken in this sort of pink shaded area. And here's the Stokes shift function from 4 representative slices. And we see there's sort of this initial decay component, but there's also these oscillatory components here. So we incorporate the oscillatory components and we extract the ultrafast time scale that we're fitting with a Gaussian. And so what we see is that now the time scales depends on the excitation frequency, right? And so to sort of look at this in a different way, I'm going to plot these time scales that we're extracting as a function of the excitation frequency that they were extracted from, right? So this yellow line matches here. This pink one is the time scale here, the 52 femtoseconds. This green one is here, and this dark green one is here. Okay. And so when we look at the time scales this way, what we find is at the center of the peak, we can extract the fastest time scales. All right, so we're extracting the fastest sort of relaxation timescales. If we look at higher frequency, on the higher frequency side of the peak, so over here, where you see a slower, timescale slow a bit. And if we look at this lower, the frequencies extracted from this side of the peak, the timescales get even slower. And so we were thinking about this, and to sort of help understand what's happening with these timescales, I want to go back and just sort of remind you of how we're extracting the Stokes shift from the 2D spectra, right? We're taking slices here, we're looking at this asymmetric line shape change, and from that, we're extracting this frequency. And we're assuming that this frequency that we're extracting is reporting on this energy gap. So if there's any other process in the 2D spectra that has a asymmetric line shape change along the omega-3 axis, the detection axis, it could influence this frequency that we're extracting. All right, so there's two processes that we were considering. One's vibrational relaxation and one's spectral diffusion. So for vibrational relaxation, right, we don't resolve all the different vibrational peaks here, but we know they're here. We can see the oscillations, right? So there's vibrational relaxation, and that would lead to a growth here below the diagonal at these higher frequencies. So vibrational relaxation, which I'm indicating as the number 1 here, could contribute to these time constants. There's also spectral diffusion, right? And so this is just a Kubo line shape for a single transition. And spectral diffusion will lead to amplitude shifts in the 2D spectra, right? So at higher frequencies, we'll see an amplitude shift to lower detection frequencies. And at lower excitation frequencies, we'll see an amplitude shift to higher excitation frequencies. And these amplitude shifts due to spectral diffusion could also affect this parameter that we're extracting, which we're taking as like from the full width half max of the transition. Okay, so spectral diffusion could also contribute to these timescales on sort of the lower energy side, right? So looking at these frequencies, but also the higher energy side. And so from these studies, what we find is that the center frequencies have minimal contributions from the vibrational relaxation and the spectral diffusion processes. And so that these center frequencies are reporting more on the solvation dynamics and have less contributions from other dynamical processes. Okay, so to summarize, we extract the dynamic Stokes shift spectra from the 2D electronic spectra with prominent vibrational coherences. And we were able to extract the Stokes shift functions, so the solvation dynamics, from slices taken at single excitation frequencies. But in sort of using this analysis procedure, we want to really consider what other processes could be contributing to this asymmetric line broadening, right? So we can do it, but I think we have to be careful. Okay, and so the next steps, we want to see how the dynamic Stokes shift really compares to other line shape analysis procedures. And so we started simulating 2D electronic spectra, right? This was motivated by sort of the shutdown of the labs, but we simulated the 2D electronic spectra of using different Stokes shifts, so the static Stokes shifts, to see how different line shape analysis procedures compare when we have a smaller Stokes shift versus a really large Stokes shift. All right, and we hope to submit this manuscript soon. Okay, and with that, I really have to thank my group, right? Here's my students. Then they really are in the lab. They're getting the data, collecting the data. So they put in a lot of hard work. And today I was only able to talk about work from 2 students. So I talked about Xiaowei's work on Crescel Violet, and I talked about Yimin's work looking at the both DIPI molecules and Photosystem 1. And I have to thank our collaborators for supplying us with these molecules. I have to thank funding agencies for supporting the work. And I also have to thank you for sticking around to hear about some of the work we're doing in my group.
Thank you very much. So the talk is open for questions. I think there's a panelist.
I have a quick question. Nice to see your talk, Jessica. Nice to meet you. It's really interesting to see how different processes in a 2D spectrum sort of add up, and how you can separate out a bit the Stokes shift from spectral diffusion and vibrational relaxation. So I was wondering if you were to just project and have a pump probe spectrum, how much would that modify the observation of the time scales of the Stokes shift that you see? So you get a separation, beautiful in 2D IR. So how does that, to what extent does that improve on the pump probe measurement?
So for the pump probe, well, we did look at the projection and we compared so that within the error bars, we might get something a little bit faster, but let me see if I can show it to you. Okay. Oh, wait, maybe I'm not sharing now. Sorry. Okay, can you see my screen now?
Yes.
Okay, cool. So yeah, so we did compare that, right? So this is the fit from the projection is sort of in this column here. And this is when we start to extract the slices. And so what you can sort of see here is when we, and what we were doing here was comparing if we incorporate the oscillatory modes or if we don't incorporate the oscillatory modes, was sort of the motivation of this paper. But what we find is that they're all sort of fast, right? They're all below 100 femtoseconds. And so when we look at, it's better to incorporate the oscillatory mode. So let's compare that one. So here we get a Gaussian time component of about 47 femtoseconds. And when we get to the fastest components that we extract in the 2D spectra, they're around 40 femtoseconds. So it's not a huge change, and within error, it's similar. I think the advantage here is that so we can extract the solvation dynamics from a single slice, but in a sense, we still want to look at all of the slices, because when you start to compare them, this may offer a means by which to sort of separate inertial solvation dynamics from sort of vibrational relaxation. And that's sort of one reason that you might want to sort of separate this out. But for this molecule, there's not a huge difference in looking at the projection. It might be molecule specific too.
Yeah, it's definitely better to know that there's not a big difference.
Yeah.
Thank you.
Yep, thanks.
Chris,
Okay, yeah,
had a question
Chris,
as well?
please go. I saw you.
Jessica, yeah, Jessica, really beautiful talk. Thank you. I especially, of course, like the line shape analysis work. And one of the things, particularly when you start comparing things like CLS and Stokes shift, one of the things I'm always worried about, even though Mark Marincelli tells me I shouldn't be, right, is the assumption that you can extract the spectral diffusion dynamics out of the Stokes shift is really dependent on the sort of linear response assumption of the excited state perturbation, right? But when you get really big Stokes shifts, that's precisely when you ought to be worried about the breakdown of that linear response assumption. And I wonder if you can tease out any evidence for the breakdown of linear response. Are the excited state dynamics really going to be different from the ground state dynamics? And can you begin to piece those differences apart?
Yeah, so that's really, that'll be really exciting, right? And so one thing, because we have the 2D IR spectrometer, right, we're able to actually do that. So we can say, okay, this is what we get when we just look at the equilibrium dynamics. But when we photo excite this, we know we make this huge change in the charge distribution. So we're excited to do those experiments. We haven't done them yet, but that'll be really cool to do, especially for two systems that are similar but might have different Stokes shifts. So that's really cool. Yeah, thanks for the suggestion.
Maybe I would like to continue that line. In body P, isn't it so that there is a charge transfer?
So it depends on your BODIPI.
Okay.
Right, so you can get BODIPI that have cyano groups on them, and then they have charge transfer. So with these BODIPIs, okay, so the Bo-Dippy that I showed has hydrogens on it. We also have other Bo-Dippies that have iodine, and there we see inter-system crossing, but we don't see evidence of charge transfer there. But it depends on the substituents, which is why we're interested in the Bo-Dippies. You can do so many, well, if you have a collaborator like Dave Chenoweth, you can get all of these different Bo-Dippies that have all of these different substituents on them, so you can look at different processes.
Okay, yeah, I am also doing a little bit on BoDP. That's why I was asking. Maybe I will ask you more questions a little bit later. I think Hoshi Yang, you were the next with your hand raising.
Oh, thank you, Maxime. Hi, Jessica. Nice to meet
Hi.
you. Yeah, it's very exciting and nice work. And can I ask some questions about the PS1 work?
Yeah.
Very interesting stuff. First question was like, do you see any annihilation processes in your 2D DAS? And another question is, for your data on the sub 50 femtosecond DAS, right? I'm just wondering what comments you would have, because like I guess we scanned T1 up to like 100 femtosecond. In other words, like 50 femtosecond is much shorter than your FID. So how much confidence in actually extracting the sub 50 femtosecond dynamics? And also I noticed that your DAS is, you probably explained, but I probably didn't catch it, but it's just one negative peak and there's no positive peak, or the other way around. So in other words, there's not so much of an energy transfer. I wonder how short are your pulses, and is there any kind of like chance of like some pulse effect that's actually being like that's kind of like interfering in your signal? Yeah, thanks.
Yeah, so yeah, those are all really good questions. Thanks. So and I think we can answer them through a lot of control experiments. So for the 2D ES, right, it's so even with the pump probe experiments on Photosystem 1, you have to be careful with the annihilation, right? Because you have 300 chlorophyll molecules, they're really tightly packed, and you need enough energy to put in to get a signal out. But if you put too much energy in, it looks like you have a really nice signal, but then your lifetimes are just really fast. And so what we did is we actually did in the supporting information a lot of control experiments with the power control. So we changed the pump pulse power and looked at pump probe spectra, and we figured out, all right, we basically made a plot to see when do we stop having linear sort of behavior with power. And so that's how we made sure that we didn't have annihilation. And I think we even put in the 2D ES power, but we only did that at 2 different powers because it's a lot more complicated experiment. So with the power, yeah, it's something to be really careful with, but doing those preliminary or initial measurements is important to make sure that you're okay. So okay, I think I wrote this down. And then with the 2D DAS analysis, so one thing that we were considering with the 2D DAS, right, because we were worried about sort of the initial, like, how do we know that this just isn't characterizing the pulses, right? And so we did a lot of different experiments where we looked at the tuning of the incoming pulse to see how this would influence the 2D DAS, and we see the same sort of 2D DAS that we're extracting. And then I'm forgetting some of the questions. Oh, we also looked at, there's also a question with the, with only seeing a positive peak, right? And so for that positive peak, we thought a lot about that, right? Because usually if there's energy transfer, you would see a decay and a growth, right? Or if there's equilibration, we see that 4-peak pattern. And so that's where we started really thinking about sort of why this could be the dynamic Stokes shift. Because there, right, your stimulated emission is just moving. It's not necessarily leading to a decay. And so the stimulated emission could just lead to sort of that growth below the diagonal without a corresponding decay. Because you have your ground state bleach stays there, and so you sort of have that shift in the frequency. And so given that the chlorophylls have a Stokes shift of approximately, well, you know, right, it's a very small Stokes shift for the chlorophyll molecules, right? And so if there's just a slight relaxation like that, I think that you could actually have that without the decay. But we can test that with a model. That would be pretty, yeah, we could really test that with the model, looking at I guess with the models that Xiaowei's doing now, for the dynamic Stokes shift.
So
Yeah.
if it's due to this Stokes shift, the absorbance will move, right? But
Right.
something else would compensate for, because it's moving. So there should still be a negative signal to replace the, the hole, if you want to call it that way, that's being shifted away. So there should still be some sort of negative signal, wouldn't there?
Right. I'm guessing what I was thinking is that if the Stokes shift is so small, how would that present in the 2D DAS? Would you really resolve the negative and a positive type of peak there? I mean, with the Cressel violet, when we do the global analysis, we see a clear sort of behavior that way. But there we have a Stokes shift of about 700 to 800 wave numbers. So the question is how much of a growth do you see there? And how much of the decay component if it's just 100 wave number Stokes shift?
Okay, got to think about it. Thank you.
Yeah.
Okay, Jacques Armstrong, please. Yes. You have to unmute yourself. Yes.
Okay, got it. I really liked your talk, and I'm kind of interested in the differences between the spectral diffusion dynamics that you mentioned are contributing to the line width changes, and also the dynamic Stokes shifts informing on the solvent dynamics. So my understanding is that they would both be related to kind of the motion of the solvent and the floppiness of the chromophore. So do you have any sense of which kinds of motions are associated with which kinds of contributions to the spectrum?
But like, well, in the sense that I think this relates back to Chris's question, right? In the sense that linear response holds, then the Stokes shift function is really reporting on the same exact correlation function that that the spectral diffusion is also reporting on. And so in that sense, they should be the same, right? But that's one of the questions is can we sort of investigate the breakdown of linear response, right? So you could have this sort of, the hindered rotational motion would be like the inertial response, that, but then you could also just have sort of the diffusional motion, like you have the making and breaking of hydrogen bonds and just methanol too. Yeah, I'm not sure, did that answer your question? So
Yeah, I think so.
cool.
I think Daniel Turner also had a question.
Thanks, and good to see you again, Jessica, and thanks for the nice talk. Relating to one of the previous questions, the first part of your talk on energy transfer and the second part of your talk on the dynamic Stokes shift are quite related, because not only are the individual peaks having a dynamic Stokes shift, but also the energy transfer peak should be having slightly different and a little bit longer dynamic Stokes shift. And I'm wondering if you've characterized that using your data. We sort of pointed to it in our previous paper, but obviously we didn't have the data, and you do. So I'm wondering, have you analyzed that to look for the redshift of your energy transfer peaks?
We haven't. So the energy transfer peaks in photosystem 1 is what you're referring to?
That's right. They should be redshifting by whatever the dynamic Stokes shift is of the acceptor in both of those systems. And so
Yep.
this, you'd have to modify the analysis to accommodate the redshifting of the peak.
And so we haven't looked at that, but that's something to look into further. For just like the regular chlorophylls in the antenna, I'm not sure we would be able to resolve it given how broad the peaks are in the 2D DAS. But what's interesting about these red chlorophylls is they actually have more of a Stokes shift for the red chlorophylls, and it's hypothesized that they might have some charge transfer character too. And so I think it would be really interesting to look at those. And we actually have some other PS1 complexes that we're looking at that have different types of red-shifted chlorophylls, and they're more spectrally isolated. So that would be a really interesting thing to look into. Yeah, and so when we get there, perhaps I'll reach out to you too, again, to talk about that a bit more.
Would be to look at the fluorescent spectra, and hopefully you get like a nice separation between the absorption and the fluorescence, and it should be there. And that would be like a perfect way to isolate the energy transfer from all the other things going on, because it will have a characteristic red shifting timescale.
Thanks.
Anyway, thanks again for the nice talk.
And maybe here then a follow-up question. Did you, when you look at the stock shift, okay, is the final, so which means the long time position of the stock shift, does it correspond to the steady state emission?
No, because for the Bo Dippy, it's more to the green. So we had it's harder to make a NOPA that broad. So we were limited by the pulse width, right? So we only had sort of some overlap with the absorption and some with the emission. So now we have a white light continuum. So we can do 2D spectroscopy where we pump with our NOPA, but we can probe with a white light continuum. And I think it'll be really cool to go back, because then if you're right, if we could actually just monitor that shift, like the frequency of the emission, then we don't have to worry about line shape analyses, right? We can just take the maximum of that stimulated emission. And so that's one area where we're interested in moving. But with our current setup, we had sort of like the same NOPA was generating our pump and our probe, so we were limited by bandwidth.
Yeah, I noticed that your pump is broader, the spectrum is broader than the probe.
That's because we're going through a dazzler too, so that makes
okay.
So we pre-compensate the pulse before going through the dazzler, and so the gratings sort of shape the spectrum a bit. So they're not exactly the same, but yeah, they're similar.
Okay. Jose Young, do you still have a question, or you have forgotten to lower your hand?
Yes, sorry. I forgot to lower my hand. Yeah, it's aching now.
Okay. More questions? Not that I see. Nothing in the chat today. Okay, well, last chance for asking a question.
Can I shoot a last question then? Because you're looking at these two different cyanobacteria, and you see that there are differences in their behavior. Maybe not huge, but there's some differences in the transfer. Is there any evolutionary or any functional reason for this, or is it just doesn't really matter for the individual bacteria? It has found maybe a slightly different solution to the same problem, or it's the essence of these proteins are from the same background, of course, so they might not be. It's just some variation that doesn't really affect their function.
Yeah, so that's a really good question. And until like recently, I would say, well, we don't know, but there's some really nice work coming out now looking at photosystem 1 from cyanobacteria, where they start to grow the cyanobacteria in what's called the far red light. So growing cyanobacteria at 700 nanometers, or between 700 and 800 nanometer light. Right. And so there you don't have the antenna chlorophyll. So you're really relying on sort of the red chlorophyll absorption for the cyanobacteria to grow. And what they find is that the cyanobacteria, they completely restructure their light harvesting antenna, and they start making different chlorophyll molecules. And so they make chlorophyll F molecules. And chlorophyll F, different than chlorophyll A, it has sort of like a carbonyl group. That leads to a shift of about 30 nanometers. And so the recent sort of structural characterization of cyanobacteria with chlorophyll F indicate that these really far shifted red chlorophylls are actually in some of these red chlorophyll sites in photosystem 1. And I think with this information, it indicates that there might be some sort of reason that these sites are chosen. And so, but before that sort of structural information and that connection with these sites being sort of specific to the red chlorophylls, I would say that's hard to know, right? But it's interesting that they are species dependent, right? If you look at the spectrum, the antenna chlorophyll, they're very similar, or the bulk chlorophyll. But when you start to get to that red shifted region, you see these changes. And so their role in light harvesting, right? Whether they play more of a photoprotective role or sort of concentrate the energy somewhere, that's still, I think, an open question. But they definitely change the absorption, right? They increase the absorption cross section to the red. So.
Okay. Thanks. Okay. I think there's no more questions.
No.
So maybe we should thank Jessica, and we should also thank Yoshitaka, but I think he already went to bed, so it's very late in Japan. So we're looking forward to see everybody again 2 weeks from now.
Thank you.